series. As a housekeeping rule, I would like to request you all mute your microphones during the presentation. The presentation will run for 30 minutes. And after the presentation, 15 minutes will be available for the question and answer sessions and discussion also. The session is being live streamed on social media and recorded for educational purposes also. Without further taking more time, I would like to welcome and introduce our speaker today, Dr. Navraj Dhami. He is Assistant Professor at Pokhara University, Nepal. He has completed his MSc in Biotechnology from Wageningen University, the Netherlands, where he got Netherlands Fellowship Program to complete the course. Recently, research work entitled Cloning and Characterization of Cyclotropin Lactone Synthesis from Inorganic Britannica lineage. After his graduation from Europe, he continues his assistant professor position at Pokhara University. And in 2015, he got higher degree research tuition fee waiver scholarship at Western Sydney University in Australia, where he completed his PhD program. His PhD research work entitled Environmental and Development Regulation of Carotenoid Metabolism in Herbaceous Leaves. He worked on numerous research topics which led to the publication of his work in high-impact journals. His research interest includes carotenoids, dietary nutrition, plant secondary metabolites, medicinal plants, plant biotechnology, etc. We are very pleased to listen to Dr. Navraj Dhami's presentation today, and I would like to request Dr. Dhami for his talk.
Thank you very much, Ashish, for nice introduction. May I request you to allow me sharing my screen, please?
Can you share your screen now?
Thank you. All right. Is it visible there, my screen?
Yes, it is.
Thank you so much. As Ashish introduced to me, my basic interest in my research area is plant science, basically plant secondary metabolites. So I'm thankful to Biomedicum seminar series organizers, especially the team from Research Institute for Bioscience and Biotechnology, particularly Prajwalji. Thank you very much for inviting me for this particular webinar. I'll be talking about regulation of carotenoid biosynthesis in leaves, particularly today. So I'm representing Pokhara University as a center. All right. I'll be talking about general overview of carotenoids, what they are, and why carotenoids are important for plants as well as other organisms, including human beings. And I will be emphasizing more on carotenoid biosynthesis in plants, particularly in leaves. And I will touch how carotenoid biosynthesis is regulated in response to different environmental interventions. So most of the work I'll be presenting here is from my PhD, which I completed in Western Sydney University, Hawkesbury Institute for the Environment, Australia. And at this moment, I would like to remember my supervisor panel members, Dr. Christopher Cazunelli, my principal and lead supervisor, and Professor David Tissue and Professor Barry Foxes, who supervisors of my panel. So I'm sure many of you, or almost all of you, have seen so many colorful plant parts or plants around us, like carrot or maize or marigold flower, tomato, capsicum, persicum, oranges. All these plants, or all these parts of those plants are brilliantly colored, that is because of carotenoids. And they are accumulated in high amount in storage tissues like root or fruits or seeds. However, they are also synthesized in leaves. However, the color of carotenoids is not visible in leaves because of high amount of chlorophyll. But leaves also contain abundant amount of carotenoids. So these carotenoids are 40 carbon containing organic compounds. They are particularly isoprenoids and they are lipid soluble. And these carotenoids are very specific in terms of biosynthesis. Not all organisms can biosynthesize carotenoids. Only the carotenoidogenic organisms, which are basically plants, algae, cyanobacteria, some bacteria, some fungi, and only few insects, 3 to 4 species of insects, including bee aphids and spider mites, only can synthesize carotenoids in vivo. And all non-carotenoid organisms, including human or animal, particularly organisms from animal kingdom, they need carotenoids, but they acquire these carotenoids from other sources, particularly plants or algae or fungi, whatever. For example, you can see here salmon with brilliant red color due to astaxanthin, either lobsters, prawn, or flamingo, or garden finches. They all saw some sort of coloration that was because of carotenoids. Our eye, particularly the macular region of our eye, that needs carotenoids to function properly. And very interestingly, if you see the ovary, section of ovary that contains a huge amount of carotenoids, particularly lutein. Corpus luteum, you might have heard this tissue in the ovary that is full of carotenoids, which is lutein. So, brief note on the history of carotenoid bases. First of all, almost 200 years back, Wackenroder, a German scientist, isolated a carotenoid called beta-carotene from carotenoids for the first time. After a century of Wackenroder's work, Paul Carter isolated beta-carotene again, and he actually identified, confirmed the structure of beta-carotene and vitamin A, which can be obtained from beta-carotene. Structure of both the compounds was elucidated by Paul Carter in 1932-33. And later, at that time, this study was really a breakthrough study. As a result, he received Nobel Prize for that particular work in 1937. That was the first event where chemical structure of carotenoid was determined, confirmed. And till the date, there is one website called carotenoiddatabase.jp. That website has enumerated more than 1,200 carotenoids obtained from different organisms, more than 700 organisms. Besides 40 carbon carotenoids, nowadays there are a large number of carotenoids reported which contain more than or less than 40 carbon as well. For example, 50 carbon containing carotenoids, 35 or 30 carbon containing carotenoids. So carotenoids are isoprenoids, lipophilic isoprenoids, and they mostly they have 30 carbons, but they might have 50 or less than 40 carbons as well. So these carotenoids are quite essential, both for plants or organisms that synthesize carotenoids in vivo or non-carotenogenous organisms that obtain carotenoids from the food source or dietary sources, including humans. For example, beta-carotene, it is a precursor of vitamin A. Vitamin A is essential to maintain eye health, eyesight particularly, and vitamin deficiency may cause several malfunctions in the body, particularly it may cause blindness. there are several breakthroughs in the field of enhancing carotenoid accumulation in plants, particularly in food crops. One of the breakthroughs is the production or development of golden rice. In golden rice, normal rice doesn't accumulate any carotenoids in the endosperm, the part we eat as a rice. However, scientists they develop genetically modified rice that can accumulate, that can synthesize and accumulate beta carotene in the endosperm. And that is why the color of the endosperm becomes orange in color or golden in color. And so the name is golden rice. Lutein and zeaxanthin, as I mentioned earlier, they are important key carotenoids present in the macular region of our eye. So these compounds, these two carotenoids are essential to protect the eye, particularly protection to protect the eye from the age-related macular degeneration. And these carotenoids are actually very brilliant quenchers of reactive oxygen species, and they detoxify those reactive oxygen species, particularly during the stress conditions. Adequate amount of carotenoid intake is essential. There are several reports already published. So we can see that this carotenoid level in our plasma or our body is related to several diseases, particularly related to cognition functioning as well as cancer. So optimum amount of carotenoids is essential to be maintained within our body. So regarding the roles of plants, roles of carotenoids in plants, as I mentioned earlier, carotenoids are primarily synthesized in plants, algae. And these plants or algae, they are photosynthetic organisms, autotrophs. And these autotrophs do photosynthesis. And for photosynthesis, carotenoids are the pigments that assist major photosynthetic compounds that are chlorophylls. Chlorophylls absorb photons from the light, but they have a limited range of photon capturing. That range is extended by different carotenoids. Carotenoids can absorb blue-green, can absorb photons or light from blue-green zone, which cannot be absorbed by chlorophylls. That is why carotenoids can enhance the range of photon absorption. Another two carotenoids, we call them xanthophylls, anthraxanthin and zeaxanthin. They are key components of xanthophylls cycle that occur in leaves particularly. And these compounds protect leaves and photosynthetic apparatus from the stressing conditions, particularly under high intensity light and high heat conditions. These xanthophylls protect chloroplast as well as the functioning of chloroplast. And besides, there are several functions yet to know. There are many things yet to reveal. Besides assistance in photosynthesis and photosynthetic functions, there are some other indirect roles of carotenoids, which are basically carotenoids are precursors of key phytohormones. Two important phytohormones are abscisic acid and strigolactones. Abscisic acid is one of the key hormones, phytohormones, that primarily regulate the stomatal movement. And through stomatal regulation of the stomatal movement, stomata closure or opening, abscisic acid can function in stress tolerance in plants, particularly under stressed conditions, drought primarily. And besides, carotenoids, particularly beta carotene, is a precursor of several stigolactones. Stigolactones are another key hormones that have a wide range of roles. The major role of stigolactones is to determine plant architecture. It determines root and shoot branching primarily. And besides, several stigolactones they work as signaling molecules, and they signal soil microbiota, particularly arbuscular mycorrhizal fungi. They attract, they stimulate mycorrhiza to grow and to flourish well around the rhizosphere, within the rhizosphere, and that indirectly support plants for proper growth and development of nutrient acquisition. Besides these direct roles, carotenoids are also precursors of several signaling molecules, for example, beta-cyclocitral, beta-ionone, etc. They are environmental signaling compounds. They respond to the environmental change conditions. So these are some general functions and overview of carotenoids. Now, I would like to focus on carotenoid biosynthesis. Carotenoid biosynthesis is pretty similar in almost all organisms. In eukaryotic organisms, I would say, in algae and higher plants, carotenoid biosynthetic pathway is pretty similar. However, in bacteria, pathway is similar, but the steps are very few in comparison to higher plants. You can see here on the left, a carotenoid biosynthesis starts from the condensation of two molecules of zeroyl diphosphate, which is 20 carbon compound, and two molecules of ZZPP condensed together in the presence of phytoene synthase, and they produce phytoene. Phytoene is the first carotenoid in the pathway, which is 40 carbon. And in case of plants and algae, there is a series of enzymes that convert phytoene into all-trans or tetra-cis-lycopene, all-trans-lycopene. This all-trans-lycopene is the major compound, major carotenoid that is accumulated in tomato fruit, ripened tomato fruit. And in between phytoene and all-trans-lycopene, these are all cis carotenoids. These cis carotenes or cis carotenoids are absent in bacteria. In bacteria, instead of several enzymes here, bacteria comprise one CAR-Tri enzyme that can convert phytoene directly into all-trans-lycopene. So this is the difference in prokaryotic as well as a eukaryotic biosynthetic pathway. However, in case of plants, this is the standard carotenoid biosynthetic pathway. And after the all-trans-lycopene, carotenoid biosynthetic pathway goes on, and it bifurcates into two branches. One is alpha branch and another is beta branch. And beta branch produces beta carotene, and then cryptoxanthine, zeaxanthin, anthraxanthin, violaxanthin, up to neoxanthin. Whereas alpha branch end up with the lutein. Lutein is the major compound accumulated in marigold petals. And beta carotene is accumulated abundantly in carrot roots and several other things. And this is the synthesis, and most of the carotenoids, these carotenoids are accumulated in very tissue-specific manner. However, in case of leaves, as leaves are places where photosynthesis takes place, so leaves comprise photosynthetic carotenoids, or carotenoids that involve in photosynthetic photon capturing. And basically, leaves accumulate, chloroplast accumulate chloroplast of the leaves accumulate beta-carotene, lutein, bioxanthine, neoxanthine, anthraxanthine, and zeaxanthine. These 6 carotenoids are accumulated in leaves, but not as high as the storage tissues like tomato fruit or marigold petals or other storage tissues. And as I mentioned earlier, these compounds, carotenoids, are accumulated very specifically in specific tissues. For example, carrot roots accumulate beta carotene abundantly. Some fruits, capsicum fruits, you can see a very interesting difference in color here. Same capsicum with 4 different colors, and they accumulate 4 different carotenoids like brilliant red, capsanthin, then lutein, beta carotene, and abundant color chlorophylls in there. So it means carotenoid biosynthesis is kind of universal. However, accumulation of carotenoids is very specific, depends upon the tissue type. So now I would like to focus on leaves, particularly how carotenoid biosynthesis takes place in leaves and how it is regulated under different environmental conditions. Leaves are dynamic plant parts, plant organs. They have different functions. It is one of the very vital parts of the plants, and it performs photosynthesis. Or we can say leaves are the organ that determines overall growth and development of the plants. However, if we just zoom into the plant leaf development, we can see 2 distinct differences as leaves grow. These are the leaves from Arabidopsis model plant. This is the plant that I worked with during my PhD. So plant is tiny. It produces like 20-30 leaves under long photoperiod conditions, about 20 leaves, let's say. And the leaves on the bottom are from the 4 weeks old Arabidopsis plant, and they are arranged according to their age. Like 0 are the cotyledons. Number 1 is the first leaf emerged from right after the cotyledons in the plant. Then leaf number 16 is the latest leaf, or we can say here the youngest leaf of this plant, and leaf number 1 is the oldest leaf here. So there are 2 types of developmental gradients in here. One convention is leaves are categorized. The early leaves are categorized as the juvenile phase leaves. Okay, this I must change this. This is not adult phase leaves. These are juvenile phase leaves, and these one are adult phase leaves. It means leaves are emerged when plant become adult. These are adult phase leaves, and these leaves are old leaves, and they are juvenile phase leaves. Means leaves emerged when plant was juvenile. And these are the leaves that were emerged when plants become adult. So leaves can be categorized juvenile phase leaves, and these are the adult phase leaves. At the same time, these leaves are old leaves, mature leaves, and leaves on the right are younger leaves. So I'll be talking more into how carotenoid biosynthesis is regulated in these younger leaves and older leaves, or you can say juvenile phase leaves or adult phase leaves. So while talking about leaves are the organs that directly come to the contact of several environmental conditions, biotic as well as abiotic. So leaves have to face herbivores, pathogens, and development of leaf and leaf functions depends on the soil nutrients, availability of soil nutrients. As leaves perform photosynthesis, leaves have to interact with atmospheric CO2 level of atmospheric CO2. Again, leaves are the organs that interact with water conditions, water level, and temperature, sunlight or light intensity, quality, day and night, all those sort of things. However, although leaves have to tackle with several biotic factors to maintain its functional integrity, leaves maintain very strictly their functions. So if we talk about the particular environmental conditions, it could be light, it could be herbivores, it could be pathogens, the temperature or whatever. Every environmental aspect, every environmental conditions affect leaf functions. And leaf functions may, impact of environmental conditions on leaf functions may vary depending upon the plant, depending upon the age of the plant, and other associated factors as well. Here I have an example with atmospheric CO2 conditions. You can see here, if we increase atmospheric CO2 level, it can alter almost all of the biological activities within the plant, within the leaf. You can see here, this is the summary from literature. So when we increase atmospheric CO2, it decreases stomatal density, stomatal conductance, transpiration rate, photorespiration, and so on. However, elevated CO2 can enhance rate of photosynthesis, carbohydrate biosynthesis, the rate of respiration, biomass production, chloroplast density. However, there are reports that explains that elevated CO2 can have, a dual role in terms of secondary metabolites, particularly isoprene, volatiles, terpenes, carotenoids, flavonoids, anthocyanins, all those things. Some literatures, they mention that elevated CO2 can enhance these chemicals compounds. Some have highlighted that they decrease these compounds. So one of them is carotenoids. That was my interest. And I checked more details into the impact of elevated CO2 on carotenoid, particularly leaf pigments, carotenoids, as well as chlorophylls. This plot is quite interesting. If you see, I tracked more than 20 literatures from the internet, from different reputed journals, and traced into the effect of elevated CO2 on the level of carotenoids and chlorophylls. You could see here, in case of both pigments, some reports, some research, very good research, they have mentioned, they concluded that elevated CO2 can increase the level of carotenoids and chlorophylls. Others, the majority, they have concluded that elevated CO2 decreases the content of carotenoids as well as chlorophylls, and some mentioned there is no change at all. So this was quite confounding, a controversial result from different research from the different parts of the world. So this was kind of challenge for me to sort it out, what actually happens in case of elevated CO2. And I used Arabidopsis to sort this puzzle out. So I'll be talking more on how excuse me. Could you mute, please?
Hello. Could you mute?
All right, thank you. So now I'm focusing on the key questions for discussion. So I'll be focusing on basically how the developmental phase of the leaf that I mentioned earlier, juvenile phase or adult phase, or the age, young age or older age, affect the carotenoid content, and how carotenoid biosynthesis, as well as the catabolism, degradation, is regulated in young and old leaves or different leaf types, And finally, do all the leaves adopt similar strategies to maintain carotenoid pool in response to environmental conditions? So I'll be showing the results from my own research to answer these questions. So first of all, level of carotenoids vary in Arabidopsis rosette leaves. If you remember previous figure, this one, elevated CO2 has a controversial impact on pigments. So I request you to remember this plot. So in here, these are again the leaves from Arabidopsis. Juvenile phase leaves are there, adult phase leaves are there, and adult phase leaves are obviously young leaves, and juvenile phase leaves are old leaves. So first, I measured the carotenoid content in these different leaves to see whether these leaves accumulate or synthesize or accumulate equal amount of carotenoids or not. The results are here. This was quite interesting result. So when I compared carotenoid content in leaves of different age or different phase, you can compare both. You could see here, leaf number 3 comprised least amount of carotenoids, and leaf number 10, 10 plus means they are youngest leaves, adult phase leaves. They had highest amount of carotenoids. I compared these carotenoids in fresh leaves as well as dried leaves. In both fresh and dried leaves, I found that carotenoid content was significantly higher in these leaves. They are young leaves or adult phase leaves, but content was quite low in older leaves or juvenile phase leaves. But this was surprising to see. This was happened only in Arabidopsis, not in other plants. I compared several plants. One is given here, Panicum. So in Panicum, older leaves comprised substantially higher amount of carotenoids than the younger leaves. Whereas in case of Arabidopsis, it is just a reverse. In other plants as well, I compared eucalyptus leaves as well. Same trend was obtained. But in case of Arabidopsis, this was unique kind of trend that I encountered with. So after obtaining this result, I had to sort out whether it is age or the phase of the leaf that is affecting carotenoid content. So I looked into the genes that regulate this leaf phase transition. Leaf phase is something that determines the shape of the leaf or other attributes of the leaf, like petiole length, all those things, and cell types as well. So there are some genes already revealed that control developmental phase transition. And the specific genes that regulate transition of developmental phase are SPL genes, SPL proteins, squamous promoter binding like proteins. They determine phase transition. And one of them, the most important one, is SPL15. So as SPL15 gene expression level is substantially higher in adult phase leaves, but very low in juvenile phase leaves. So I was thinking whether this SPL expression level is connected to the level of carotenoid content in the leaves. For that, I had mutants in the lab, and I checked SPL mutants and the other mutants that regulate developmental phase transition in Arabidopsis. And I compared carotenoid content in these leaves, very old leaves or juvenile phase leaves, and young leaves, adult phase leaves. However, results are here. You could see younger leaves, they had significantly higher level of carotenoids. All the mutants, they had similar trend of carotenoids, similar to the wild type. So younger leaves, higher amount of carotenoids, older leaves or mature leaves, low amount of carotenoids. So from these results, we concluded that no, SPLs or phase transition has no or unknown role in regulating carotenoid content. I just left this experiment at that point and jumped into another aspect of whether the age of the leaf is the factor that controls carotenoid content. For that, I first took.
Excuse me, sir. Sorry to distract you. Our time is already over. Could you please try to shorten the presentation?
All right. Thank you.
Thank you.
I'll try as soon as possible. Then I compared other leaves, younger leaves, and again you could see here the youngest leaves had highest amount of carotenoids. And then I compared three different types of leaves: the cotyledons, youngest leaves, juvenile phase leaves, and adult phase leaves at 2 time points: day 9, day 14, day 14, day 21, day 21, day 31, and compared young and old leaves. And again, young leaves had significantly higher amount of carotenoids. From this, I concluded that it is the age of the leaf that determines the content. And second task was whether these carotenoid biosynthesis is regulated at the level of gene expression. I checked into one website, it is a database which has compiled gene expression data in there at 2 time points, 14th day and 24th day, and sorted out a couple of genes that are differentially expressed in young versus old leaves. Then I collected different mutants that were disrupted in several different genes, both carotenoid degradation genes as well as biosynthetic genes, and compared their carotenoid level in young and old leaves. Again, I found that young leaves are accumulating substantially higher level of carotenoids compared to the old leaves. So degradation is not related to the differential amount of carotenoids. Later on, I compared biosynthetic pathway mutants, and again found that this biosynthesis is not directly related to the higher and lower level of carotenoids in young and old leaves. And I looked into another aspect, that was the precursors, whether the precursors are coming at different amount in young and old leaves. And I found a couple of target genes which were differentially expressed in young and old leaves. And I then jumped into check whether the precursors are coming higher in young leaves or less in older leaves. For that, I used norflorazone assay. Norflorazone is the chemical that can inhibit activity of PDS, phytoN desaturase, and blocks carotenoid biosynthesis. As a result, phytoN is accumulated in the leaves. So I did this assay and measured phytoN accumulation in young and old leaves, and results are here. You could see younger leaves are accumulating substantially about 3 times higher phytoN compared to the old leaves. And from this result, I concluded, okay, there is something that is controlling metabolite precursor flux into the carotenoid biosynthetic pathway, which is higher in younger leaves and lower in older leaves. So to confirm this, whether the flux is higher in younger leaves, I tested plants with elevated CO2. As elevated CO2 increased the metabolic flux into the pathway, overall metabolite abundance is increased under elevated CO2. And I grew plants under elevated CO2 and compared carotenoids, and again found that younger leaves enhanced carotenoid content under elevated CO2. From this, we confirmed that young leaves are much more sensitive to environmental conditions, and metabolite flux is higher in younger leaves. And finally, I compared whether other environmental conditions, for example, darkness and cold, higher temperature can affect carotenoid biosynthesis in leaves. And yes, higher temperature or cold conditions or dark, all three affected carotenoid content in younger leaves, but not in older leaves. The conclusion is that older leaves are quite resilient to maintain its carotenoid pool. However, younger leaves are quite plastic, although they produce substantially higher amount of carotenoids. They are quite plastic. They can alter carotenoid content instantly. And most intriguingly, carotenoid biosynthesis in leaves is completely switched off during the dark conditions during the night. So to summarize, leaf age is the main factor that affect the level of carotenoids in leaves in Arabidopsis, and precursor flux is higher in younger leaves and in compared to the older leaves. And young leaves are more plastic, whereas the older leaves are much resilient. And based upon these results, we can say that younger leaves of Arabidopsis, they are quite good model to explore more into carotenoid biosynthesis and its regulation in plants. So I thank you all for listening me and inviting me for the talk. So if you have any questions or feedback, please feel free. Thank you very much. I'm sorry, I've taken a couple more minutes, almost 10 minutes more.
Can you stop the sharing, Dr. Sami?
All right.
Thank you very much, sir, for your wonderful presentation. I would like to thank Dr. Dami for his talk on carotenoid biosynthesis. It was very helpful for us and those who want to research on carotenoid biosynthesis or its mechanism. Without wasting time, I'd like to proceed to our next session for the question and answer session. If anyone from participant can ask question related to topic to Dr. Dami. Anyone, please. And you can also write your questions on chat box also.
Thank you, Dr. Thami. I have one question. Can you listen to me?
Sorry, you're mute.
Hello.
All right.
Hello. So what happens when we when we cook the leaves of this spinach, like sag, in our home? So does this carotenoid remain after cooking with the leaves of the sag or different kind of sag we find in Nepal?
Oh, very nice question, Prajalji. I did one experiment that was not actually for cooking, but I was looking into whether fresh or dried leaves they have same amount of carotenoids or not. So what I found during my experiment was some carotenoids was substantially enhanced during the drying process. Particularly zeaxanthin and anthraxanthin were significantly increased, which were almost absent in fresh leaves in Arabidopsis. While drying, they had to go, in the incubator for like 24 hours. So in the beginning, if leaves they encounter with higher temperature, they start synthesizing stress. They start combating with that high temperature, and they start synthesizing zeaxanthin and anthraxanthin. So that was what I noticed during my experiment. As carotenoids are compounds, they scavenge the ROS, reactive oxygen species. While cooking, initially leaves might enhance the concentration of carotenoids. However, once that limit goes up, they may start degradation because carotenoids are very sensitive to light as well as the higher temperature. They may undergo degradation. To some degree temperature, like 30 to 40, we can enhance the level of zeaxanthin and anthraxanthin.
Thank you.
More?
If So can we conclude our session here?
I think there are no more questions, so we can
We'll conclude our sessions here. So thank you very much once again, Dr. Dhanush, Namrata Dhanush. We'd like to interact with you on this topic on futures also. And thank you for everyone for participating in our webinar sessions. And Meena,